Hello, everyone, and we're excited to have Stephanie Dougan be here to present her work on how focused ultrasound can may be able to help defend the body against pathogens. She was a key part of our pancreatic cancer workshop in the fall. And we're continuing to move forward with some of the recommendations that she and the group made at the end of that workshop. So we're excited about that, and we're excited to hear what progress she's made in this going forward. So Stephanie, it's great to have you here. I'm sorry I'm not there directly, but we're delighted to have you join us.
Well, thank you, Tim, and thank you to the foundation for inviting me back. I want to make this a little bit open-ended and interactive. So I'm an immunologist. I actually teach a lot of immunology at, undergraduates, medical students. So happy to answer questions or explain anything that you're thinking about. I think at this point, we don't have a lot of solid evidence as to exactly how focused ultrasound interacts with the immune system. And so a lot of this is still exploratory. So I thought I would start by kind of explaining how we think about cancer immunology and how cell death processes interface with that, and how we could really study, what the role of focused ultrasound is. So there are my slides. I do like to start by reminding people the reason we have an immune system is to defend the body against pathogens. And to the extent that it does anything else is an evolutionary accident. Fortunately, we do have some evidence that the immune system can fight cancer as well, although this is not its primary purpose. So it doesn't do this particularly efficiently. We're really good at seeing virally infected cells, and we can sort of see cancer cells if they kind of look like they've been infected with a virus. And so how does this work? And so primarily what we're talking about are T cells. So these are, white blood cells in your body, but they have a particular T cell receptor that's able to see what's inside of infected cells. And the way they do that is by having the infected cell express MHC class 1, and this presents peptides on the surface of the cancer cell. And so this can be peptides from a virus, it can be peptides from the cancer cell that are unique to that cancer cell, but in the vast majority of cases, it's actually just whatever the cell happens to be making, which is your normal self proteins. And this is just saying, hey, this is me, and don't react to this. So how do we distinguish a cancer cell from a normal healthy cell is actually a huge question that the immune system deals with all the time. So this question of self, non-self, there's a lot of philosophy of the immune system. So how do you recognize what is you and how do you recognize what is foreign is an important question in immune tolerance versus mounting an immune response. And so in terms of cancer, we've thought about this for a long time. And so this is kind of a theory put forth by Bob Schreiber, saying that as tumors evolve, it's not just the oncogenic process where you have sort of loss of tumor suppressors, gain of oncogenes, but at the same time, this is evolving over, many months to years, and the immune system sees this too. And so I think we have good evidence that at least the immune system is noticing that cancer is forming. And the thought is that it could happen in sort of these three phases. Eventually, when you have cancer growing out, this is escape. So whatever the immune system was going to do, it failed by the time people present to clinic. But before that, maybe there was this process by which you had a few cells that started to go under oncogenic transformation. And maybe you had T cells or NK cells or macrophages that have come in and started to eliminate these. As the tumor acquires more mutations and is able to evade this process, there could be some period of equilibrium where the tumors are very small, but they're not quite eliminated, and then that eventually will grow out to cancer. And so how does the immune system, or how do we know this happens? This is probably the most important reference in all of cancer immunology, so I do just want to put it up here. Again, from Bob Schreiber's lab, saying that, okay, if we think the immune system is actually surveying for cancer and making sure that you prevent cancer formation, the way you show that is by saying if you don't have an immune system, can you show that you get more cancer? This has been very hard to demonstrate in humans, but at least in mice, we've been able to show that if you have these RAD2 knockout mice that can't make T cells or B cells, then and you treat them with methylcalanthine, which is a carcinogen from cigarette smoke, and you just paint it on the mouse, they get all of these sarcomas. But actually, if you don't have any T cells, you get many more sarcomas than wild type mice. So this was still our best and most conclusive evidence that T cells are important for preventing the formation of cancer. It's probably also true that they have this equilibrium phase too. And so how do we know that, as you have cancers forming, that T cells are actually doing anything at all? And so this was a similar experiment also by Bob Schreiber's group, where if you take mice and you paint them with a really low dose of this methylcalanthine, so they get a small amount of carcinogen, then some of them will still grow out tumors. You get rid of those and you say, okay, I've got a bunch of tumor-free mice. Now, maybe they're tumor-free because it was a low dose of carcinogen and you just didn't cause any cancer. Or maybe they're cancer-free because you have a low dose of carcinogen and they have very small tumors and the immune system is keeping them in check. And so what they did is they sort of measured this over time and asked where that arrow is. If we say, okay, why don't we try to remove all the T cells at this point? Can you see that the tumors grow out? And in red at the bottom, you see that's exactly what they did. So if you have a control at the top, all those tumors are still undetectable or very small. But if you deplete the mice of T cells, you show that the tumors all grow out. So T cells are important both for preventing the formation of cancer and for keeping it very small and under control. That was all in mice. I mean, how do we know that this happens in humans? It's a little bit harder to study this, mostly because we don't have fully immunodeficient people. And so if you look at, say, what kinds of cancers do patients with AIDS get, I mean, they do get increased frequency of cancer, but it's mostly Kaposi sarcoma and some lymphomas, which are caused by Kaposi sarcoma virus, right? And so then you're like, oh, is this because they got viral infection or is it because there's actually a role in cancer surveillance? And so, we have a few points here that suggest that this probably is happening in humans. I mean, the very long time to relapse that you see in diseases like melanoma or breast cancer, where you undergo surgery and then, 10 years can go by before the patient recurs, suggests that, maybe that tumor is just growing very, very slowly, or maybe there's actually some immune control that's keeping that in apparent remission for a long time. I think the most striking but kind of frightening example are instances where patients donate organs, right? And so they think they're ostensibly a healthy organ donor, but if they have two kidneys and the kidneys go to two different transplant recipients, you have to immunosuppress them so they don't reject the kidney. But then you see that melanoma grows up in both of those recipients. And so it's a very tragic circumstance, but that's actually been reported many times now, that melanoma apparently likes to hide out in the kidneys, and you can have a couple of cells there and people don't even know they have cancer until you get to an immunosuppressed transplant recipient and it all grows out. Okay, so how does this happen? How do we get these T cells? So there's, we know this is happening spontaneously. And I want to walk through this process carefully because this is the part where we really could make it better with focused ultrasound. Okay, so you have these, tumor cells or something going out here in the tissue, and you have this purple dendritic cell. This is actually the most important player here. So this dendritic cell, which is in all of your tissues of your body, are constantly picking up things. They're phagocytosing little bits of debris or tumor in this case, or lotion you put on your skin, right? So why don't we always mount immune responses to all the foreign things we see, right? I mean, this goes back to how do you know that something is your own self versus a foreign entity? If it's encoded in your genome, you think, oh, maybe we figured this out during development of T cells. But if it's the food that you've just eaten, or if it's the lavender skin cream you're putting on yourself, I mean, these are foreign substances, why don't we make an immune response? And so what happens is that at steady state, you have dendritic cells in your skin that are picking up things, they're taking it back to the draining lymph node, and they're saying, there's no problem here, this is just normal. So any T cells that are going to react to lavender or to, ham sandwich or whatever it is, are told that, hey, there's no problem, you should tolerize, right? And so if the dendritic cell gets back to the draining lymph node and it finds its T cells, the CD8 T cells are going to become anergic, and the CD4 T cells are going to become regulatory T cells. And this is happening all the time. This is why most of us don't have food allergies and don't make allergic responses to the stuff you're breathing and the stuff that you're exposed to environmentally. It's probably also happening in cancer. Cancers grow very slowly, and there's not necessarily a sign that they're infected or that there's any danger. What you hope happens, though, is the opposite, right? So if you have, say, a viral infection and you've got a virus and it has ways to activate these dendritic cells and say, oh, there's actually an infection, there's actually a problem, then instead of a few dendritic cells going back, these dendritic cells go back in large number. They upregulate their MHC molecules, and they upregulate these co-stimulatory molecules, B7-1 and B7-2. And so when they get back to the lymph node, they say, aha, like the things I'm carrying now are actually a problem. Like this is actually a viral infection or bacterial infection or something dangerous. And now when I find these T cells, you shouldn't tolerate, you should become activated. And so the cell that really makes that decision between tolerance and immune activation is the dendritic cell. The T cell doesn't have any way of seeing that. All it says is, I see this thing that's my antigen, but is that a harmful antigen or is that a normal self-antigen? It doesn't know the difference. It has to be told by the dendritic cell. So what kinds of things can be tumor antigens? And I think this has been a very rich field that's been learning about for several decades now. So, cancer arises from your own normal self cells, but it's not identical. And so there are some things that are different that can be recognized as non-self. This is just a list of categories. So sometimes these are actual mutated proteins. That one's sort of listed at the bottom. We call these neoantigens. You may have heard of this, but it's certainly not the only thing that could be different about cancer. And so depending on what kind of cancer it is, if it's a virally derived tumor, you can still have some of those viral antigens. You can get re-expression of things that are only expressed during development. And so things like telomerase could be seen. You can have endogenous retroviruses. And so although cancer is very similar to your normal self, there are certain things that can be seen as different that T cells can recognize. But again, the T cell doesn't make that decision that this thing is harmful. It just says it's different, right? And so some antigens are shared, things that are sort of, if you have melanoma and it's super black, right? All of the proteins that are involved in melanin synthesis can be seen by the immune system. And so those tend to be shared among different patients. If it's a mutational antigen, that tends to be unique. And so, if you have random mutations that have generated antigens, that's not necessarily going to be shared from person to person. All right. This is the trickier question. So what kinds of things can be tumor adjuvants? So how does that dendritic cell get activated? Right? And so the things that activate the dendritic cell are the sign that there's actually a harmful process going on. We're really good at doing this for bacteria and viruses. And so bacterial cell wall components, viral nucleic acids, we have receptors on dendritic cells that are really good at saying, this is a microbial pattern, right? It looks like a microbe. I don't really know if it's flu or if it's SARS-CoV-2, but that's not my problem. All I know is a virus, right? So, there is a reason to go back to the draining lymph node and to say, hey, there's been a viral infection here. What this is in cancer is less clear, right? And so, if you don't have a microbe present, there are still ways that you can say that there's danger happening, but these are far, far less good than the sort of microbial induced ones. And so when we think about what could be happening in cancer or in sterile inflammation, ischemic damage, so ways cells die can also activate dendritic cells. And so if you think about necrosis as opposed to apoptosis, you're releasing things like ATP, HNGB1, that are not usually found in the extracellular space. And so dendritic cells can recognize those. As you start to have sort of oxidative damage, you can get oxidized phospholipids in the plasma membrane, and those can activate dendritic cells too, or extracellular DNA. If you're thinking about apoptosis, you've nicely blubbed your nucleus and everything is sort of compacted inside. But if you have DNA spilling out everywhere, that probably was a damage-associated cell death. And so these things can also activate dendritic cells, but the dendritic cell is not totally sure. It's not sure like it's sure if it saw a virus. It's kind of like, so when it gets back to the draining lymph node, these are not as good. That's what I've put up here. Any questions on this? Sorry, I've thrown out some words. If you're not sure at all what I'm talking about, I definitely go back and revisit some of this. We have a whole hour and there's some questions, so I'm happy to explain this. This is the fundamental part we can improve. So I think if you're thinking about what can we do for cancer immunotherapy, you can't really change the antigens. Whatever that person has, that's their antigenic burden. And maybe they've got some great neoantigens. We hope they do. If they don't, going in and mutating them at this point is not going to help because they won't be clonal. But certainly we can change the adjuvants. And so there's a lot of thought into manipulating the space therapeutically. All right. And so actually, I originally rotated with Glenn Dranoff back in the day. And so I'm going to propose in situ vaccination, which has been talked about in various forms. And I think that's sort of where focused ultrasound fits into the cancer immunotherapy modality. And so the idea of an in situ vaccination is that rather than just surgically removing the tumor, you're killing the tumor while it's still in the body so that you give dendritic cells the opportunity to kind of take up those antigens and prime new T cells. And so the very first evidence that this could happen was actually from this paper in 1993, where Glenn Dranoff took melanoma cells and asked, okay, if we engineer melanoma cells to express all of the known cytokines, which at that point there were like 10. And then we irradiate them and put them back into a mouse. Can we turn that into a vaccine? And most of the cytokines he tried didn't really do that, but one of these cytokines generated completely tumor-free mice, and that was GM-CSF. Right? And so at that point, he was like, what even is GM-CSF? This is how we learned that this is a growth factor that's important for activating and differentiating dendritic cells. And so the idea was that as you had these dying tumor cells, and in the process of dying, they were secreting GM-CSF, then all the dendritic cells would come in, massively take up these dying tumor cells, and that's why it was such a good vaccine. This was applied to humans. It actually looks really good in phase 1, phase 2 trials. And the original early trials of ipilimumab were actually all patients who had gotten GVAX first. And so that was sort of where this fits into historical cancer immunology. And so there are some definite advantages to using the person's own tumor as sort of this in situ vaccine. So here, first of all, this is extremely personalized medicine. So whatever tumor antigens they're going to have, you don't have to sequence it. You don't have to know what they are. They're all there because it's that person's same tumor. It's pretty straightforward. You're trying to kill the tumor in that person. And so a lot of times we're thinking about intratumoral injection, or if you have removed the tumor, you're just putting it back in after you've killed it. This third point's a little controversial. I think this is actually how chemotherapies work when they're curative. But the major challenge to this is how do you kill these tumor cells in a way that's really going to be very immunogenic? Now, genetically engineering them to secrete GM-CSF is a very laborious process. So although this does work, this is not like the manufacturing for that has been very challenging to do at scale. So it has not made its way out of phase 2 trials. Oh. All right. This is just one example from my lab where we kind of worked on this. So this is a mouse model where we used agonistic anti-CD40 and radiation. So this was just standard SBRT. But what you can see is that we got really robust immune responses. And so not only did we cure the mouse of its original tumor, but we induced so much priming that this white patch of fur here is actually vitiligo. And so we got an immune response to the tumor, but we even broke tolerance to the healthy melanocytes and induced autoimmunity in that area. All right, so it is possible. And so this is sort of where the field is, right? Thinking about what you need for a vaccine, you need, two things for an immune response, an antigen and an adjuvant. The antigen, as I said, we can't really fix. It's whatever the person has, but the adjuvant is something we can induce. They really need to be at the same place and the same time because, again, you're trying to get, the same dendritic cell that has taken up these tumor antigens also needs to be activated. So why did that dendritic cell decide to upregulate MHC, to upregulate co-stimulatory molecules, and to go en masse to the draining lymph node? And so there are several options that I think are kind of all in this category. Oncolytic viruses can do this. And so we do have an approval for TVEC and melanoma where you can inject this modified herpes virus into subcutaneous lesions, and you get a nice immune response and clearance of those lesions. There's been a lot of work looking at intratumoral bacteria that's not yet approved, but again, the thought that the best way to activate dendritic cells is with real pathogens. So sticking an actual virus or actual bacteria has a lot of advantages to it. You can also figure out, what are the things from these microbes that really activate receptors on dendritic cells, and so TLR ligands and STING agonists are both doing that directly, just saying like, here's the molecule to really activate dendritic cells. A lot of thought that this is what radiation does, although similar to focused ultrasound, this has been a difficult thing to pin down exactly what the effects of radiation are. But certainly this in situ vaccination is one outcome. And then, my lab's been looking a lot at local delivery of cytokines, which is actually why I was at UVA and talking about this yesterday. But I think for, this working group, really thinking about how focused ultrasound can act as an in situ vaccine and how can we really measure that are important goals for the foundation for saying, is this whole immunology connection real and how do we augment it? So I made a few bullet points about how do you measure this immunogenic cell death? How do we know that we are doing something that's going to activate these dendritic cells and increase T cell priming, right? And so there are sort of several ways that you could think about this. Some of them are really in vitro assays, right? So can you measure the release of some of these damage-associated patterns directly? I mentioned the extracellular ATP, extracellular HMGB1, as they're being released from dying cells, are sort of known and accepted adjuvants. So you can measure these. You can also ask how they act on dendritic cells. And so dendritic cells are pretty easy to make, both from mice and from humans. You can get monocytes from healthy donor peripheral blood. You can differentiate them into dendritic cells. And you can say, if I dump on my dead cell products, how do those dendritic cells get activated? And you can measure directly the things that they need to show to T cells. You can measure MHC. You can measure these co-stimulatory ligands easily by flow cytometry. I like these because it really gets at exactly the thing we're trying to say, like, did we activate a dendritic cell? Everything else downstream is further and further out from what focused ultrasound is trying to do. And so although we're looking for, more T cells coming into the tumor, we're looking for tumor regression of a non-treated lesion, Those are all things that are multi-steps down the way. Whereas here you're actually asking, did we make immunogenic cell death, right? You can measure all of these steps very precisely in mice. I put up this bullet point, but actually I think the studies that you're doing with Tim already are great. And I think he's well on the way to answering this question for you. So I don't really have anything to add other than to say, good job funding him. But humans is a bit trickier. So in mice, it's very easy because we have great models where we know T cells that recognize particular antigens. And so you can say, I have my ovalbumin-specific T cells, and I'm going to put them into a mouse, and I'm going to track sort of when they get primed in the draining lymph node. I'm going to be able to find dendritic cells. We can measure the amount of the antigen on those dendritic cells because we have antibodies for it. Is somebody trying to ask a question on Zoom?
Francesco.
And all of these tools are lacking in humans. So I think mice are great models for showing you that immunogenic cell death is possible, but it's not ever going to show you that this happens in the context of a human clinical trial. So what can we do in humans is a little trickier. We certainly, these in vitro assays can be done with human dendritic cells and human killed tumor cells. So I think this is critically important to do in human systems. But in the context of a clinical trial, I think the closest we can get is to looking at T cell priming and saying, do we have a more diverse range of T cell clonotypes that we think are responding to these tumors? So just to give some pictorial examples as well. So this is looking again in vitro. You can actually measure things like HMGB1 directly by an ELISA. And so this is as simple as taking your tumor cell line, and in this case, we've treated with some chemotherapies or targeted therapies. Cisplatin has been sort of cited as an example of a chemotherapy that induces some immunogenic cell death. There was a thought that KRAS inhibition would do the same thing, but at least in our hands, it doesn't. And so here you can kind of see if you kill cells, well, the more you kill them, the more they're just dead. But if you kill them a little bit, you can get nice secretion of HMGB1 before they die, whereas adagrasib did not do that in our hands. This is, I think, probably the most straightforward thing to do quickly for focused ultrasound. And so this is really asking sort of how does the mechanism of cell death affect dendritic cells? So actually, I'm gonna spend a while on this slide and take some questions too. So you can take dendritic cells. And again, we can make these from mouse bone marrow. We can make them from human peripheral blood monocytes. You can certainly do this all in human systems. At baseline, they actually don't have very much MHC or these co-stimulatory ligands on their surface. But if you add something, now you can add bacterial products like LPS, you can add viral products like some viral nucleic acids. They activate really well, and you can show that everything on this list goes way up, right? And the question is, if you kill your tumor cells via all these wonderful mechanisms of focused ultrasound that I have learned about from you guys, I had no idea the different types of thermal ablation and mechanical ablation, whether it's like really vigorous or less vigorous, and how many machines are able to do this. And so I think, some of the issues I've heard this morning and at the workshop in November was that everybody has their own different kind of machine. And how do you know like what kind of microbubbles or what kind of thermal ablation for how long is going to be best? So, tumor destruction is easy to measure, but this effect on the immune system is a little bit more vague. But you can actually just kill your tumor debris, however you're going to do it, from all of these different machines all across the country. And you can say, okay, we're going to ship them to one central location, and we take dendritic cells. You can get, 5, 10 different donors so that you get a range of the human population. And say, if I add all of my different fuss-killed varieties of stuff, And then I look, how well did we induce these molecules, right? And you've got your, bacterial products as a positive control and say, like, does anything get us to the level we see with LPS? So that you can do with humans because you don't actually need to know the antigens. You don't even need T cells. And this is, I think, the most direct way of asking, of these different molecules that we know and all the ones we don't know, have we collectively been able to activate dendritic cells? So any thoughts on or questions on this part?
So you will need biopsies.
No,
No?
you need like a xenograft tumor, and you take that tumor, and I mean, I've seen the videos from you guys. You stick it in the
Yeah.
machine and it liquefies, and then you just put that in an Eppendorf tube and you ship it.
But if you want to do it in humans, we need to have samples of the tumors to do your organoids or.
I think that a human tumor growing in a mouse would actually be totally fine for this.
Okay.
I mean, if you had patient samples, that would be great, but they think the process of cell death is probably not so different whether it's the human cancer versus one that you've grown in the mouse. So I think this could be very easily done in the preclinical setting.
I have a question from the audience.
Hi.
In various types of investigations with in vivo humans, if you will, we do biopsies. Prostate cancer is a perfect example where we will do saturation biopsies of the prostate in order to determine where in the prostate the cancer actually lies. Is there any value in doing in vivo biopsies or aspiration of tumors that have been treated with ultrasound in live walking human beings?
Yes. I was talking with Fred about this morning, and sort of what can you do for immune monitoring in the context of a trial? And certainly the best version of that is getting paired biopsy. So you get something before treatment and then you get biopsy after treatment, and you can do single cell analysis and you can ask what changed. Some of those single cells are going to be dendritic cells. And so all of these questions you could get, including vastly more information. But it does require that paired tissue and rather expensive analyses. And so I hope that the foundation would get there. But I think there's a lot of stuff you can do that is cheaper and easier to get an answer quickly, a sort of, what kind of cell death are we looking for? That you could answer with these kinds of studies. But ultimately, I like that idea. I can't see who asked that question, but baking in paired repeat biopsies into clinical trials is actually really, really important.
And when you do these paired biopsies, do they have to come from exactly the same portion of the tumor because different portions of the tumor are going to react differently, or can they be sampled anywhere? That is, do you need a means of accurately and precisely sampling the same spot?
Well, ideally, that same spot has been liquefied, right? So you may want the spot that's sort of near it, rather than exactly the same spot you went into. If you're just biopsying a bunch of necrosis, it's not going to look very interesting. And so we see the most exciting immunology happening is on the border of the live and necrotic region, where you have sort of macrophages and T cells starting to infiltrate in. And so I think it doesn't need to be exact. It needs to be in that general region.
And do you need tissue, or is aspirate acceptable?
I'm not sure what prostate aspirate looks like. I mean, I know this more for pancreatic cancer.
No,
So
so
what-
this is
what are you
not
thinking
just in
about?
prostate. This would be anywhere. Would aspirate show what you're looking for from an immunogenic point of view?
How is an aspirate different from a biopsy?
Biopsy is actual tissue, and aspirate is where you're just mostly getting extracellular or intracellular fluid using a very small needle to withdraw.
Yeah, I
Meaning
think
it's
you
less
would need
traumatic to the
this.
tissue.
I think you would need the cells. So a biopsy would be more helpful here.
Okay, thank you.
Okay.
Can I?
Yeah.
Ask one more question about the mechanisms of ICD. So you said that the example you gave where you, treated the cells in vitro with the chemo, if I understood correctly, you said that you need to have a dose that will not kill the cell instantaneously so that they have time to die in an immunogenic fashion.
Yes.
Is it correct?
Yes.
Okay. So when we do first, I mean, the zone that is ablated, if it's an ablation modality, I mean, the cell should be instantly killed. And so it is why you think we are seeing stuff happening at the periablative areas.
No, just that if you're looking for the immune cells coming in, a month later, it wouldn't be in the area of necrosis. It would be kind of at the border. Yeah.
But coming back to the idea of immunogenic cell death, it won't happen in the area where you're treating. I mean, very, very few, because supposedly you are destroying everything, ablating everything at the treatment area, right?
Well, I think we don't really know. So, I mean, this is the issue is if you completely liquefy your cells and there's absolutely nothing left, and this isn't great at inducing immunogenic cell death, right? But it's possible that as they're dying, if you got reactive oxygen species and you made some of these oxidized phospholipids, if those are still present, that could be helpful. I don't actually know what's the best way to kill cells, but the nice thing about this kind of assay is that instead of, a clinical trial or even mouse studies where you can test maybe one or two different modalities, you can set this up in a 96-well plate, right? If you had 300 different ways, I wanted every single fractionation of temperature, degree, and time, or size of mechanical bubble, you can test that, right? And I think that that's something where you could get at some of these answers and saying, like, when we see liquefied tumor, is that good? Is that bad? Do we want it less liquefied? Do we need it to slowly liquefy? Like, how does that affect dendritic cells? Anyway, it puts some like hard parameters and things that you can quantify on a field that has been very difficult to quantify. Okay. And so back to how we would think about this in terms of humans. And so again, finding these dendritic cells is very difficult. I think if you have paired biopsies, there's a chance, but they are some of the rarest cells in tissue. And so even if you did single cell in your paired biopsies, you're maybe only going to get like 100 cells back that would fit this criteria for being dendritic cells. And so another way we can do this, which is less direct, but looking at the T cells. And so if you have sort of new dendritic cells coming, you should be able to say that more T cells with different T cell receptors expand. And so we can actually track these by their T cell receptors. It is a genetic barcode. This is a little bit of what I presented in November, but I do just want to remind people quickly that when T cells mature in the thymus, they have these random rearrangements to make their T cell receptors. And so then every daughter cell has that same genetically encoded receptor. So that if you have sort of this priming happening in the lymph node, the cells that expand, you can say all of these blue cells came from one original precursor. Right? And so I'm going to give you an example of where we've done this. This is a collaboration we have with Jiping Wang, who is very interested in focused ultrasound, and we are getting the histotripsy machine supposedly someday at the Brigham. So I think we are very eager to work with you guys. The trial that we currently have ongoing is actually Y90 radioembolism. I'm going to show you these data because I think they're conceptually very linked. I mean, we are asking questions about did sort of ischemic cell death and what we get from this Y90 radiation, did that induce immunogenic cell death and priming? It's not FUS, but it's from the way we're measuring, it's exactly the same as what we would do for FUS. And so we're saying increased T cell priming. We're going to measure this by increased diversity of the expanded clonotypes. And so the way this trial worked is we used a backbone of CTLA-4 and PD-1 blockade. This is the trial that got this approved for hepatocellular carcinoma, where the line here in blue is significantly better than the sorafenib standard of care in black. Unclear that there's a difference between the red and the blue, but in any case, nobody was testing the red, that's just there. So the difference between blue and black got the blue line approved. And so the trial that we have is a phase 2 trial. Everybody gets checkpoint blockade, and then half the patients get the radioembolism and half don't. And so this is actually a really nice setup because you have a 2-arm trial, you have a control group, and you have a group where you can say, okay, well, is the effect of the beads actually important? So everybody gets checkpoint blockade, but checkpoint blockade works better if you have T cells to reinvigorate. And so the thought was that having this immunogenic cell death would give you more T cells that would then be targets for checkpoint blockade. And so we tried to do this regionally. And so working with Jiping is great because, I get these lovely pictures from the frozen section room. And so here, we're looking at sort of regions of necrosis where you can see like there's a little necrosis there. You've got kind of best viability, and then you've got normal liver. And so I asked for all three of these pieces to say, can we see that T cells are sort of moving around? Are they going from areas of necrosis? And we also got peripheral blood. So to try to differentiate which T cells were just like random blood T cells that happen to be in there and aren't doing anything, ones that hang out and live in the normal liver and are probably not important for your cancer, versus ones that are sort of there shared with the live and the necrotic regions. And so we did a lot of single cell sorting or single cell sequencing, where you get the transcriptional profile so you can know what cells are there. You then do a second step, and unfortunately most people don't do this in trials because it doubles your sequencing costs, but I think this was actually really critical to sequence the T cell receptors as well. And so we've just started this. There will eventually be 20 patients on the trial. We only have 6 sequenced right now. So we've got a 3 and 3 comparison. It's a little hard to say. This is kind of how the transcriptional profiling looks, where, this is peripheral blood, and this really just looks like expected. I mean, I don't think that anything changes about the global T cells that are in peripheral blood. And if you ask about sort of what kind of T cells there are, there's not really any change with treatment, which is totally fine. And even if you look in the tumor and you ask, is there a difference in the kinds of immune cells that are there? There's also not much of a difference. I mean, we can see, normal liver looks different from tumor, but whether or not you got the beads does not actually change anything. Again, kind of as expected, because we're really thinking that the biggest change will be on T cell priming, not on the macrophages or neutrophils that are there. And so this is really the important part of the study, where we asked about TCR sequencing. And again, from one T cell, you can get massive expansion. And so sometimes you see the scale goes up to 900. So whatever you see in red here, that's at least 900 different T cells that came from one parent cell. And so when we see that, we say they're probably expanding for a reason. Like those are more likely to be your tumor-specific cells. And so you can see TCR expansion in blood, TCR expansion in tissue. Well, the ones in blood aren't necessarily tumor reactive, but the ones in tissue probably are. And so we've done all sorts of matching. These are ones that are in the tumor, but matched to peripheral blood or matched to the different tumor samples. And you get these Venn diagrams where you can say, okay, if I look at which T cells are present in blood, in the necrotic region, in the normal liver, and in the region of best viability, to where are the areas of overlap where we think those would be most interesting, and sort of do we see an effect of the beads on expanding those numbers? And so kind of boxed here in red are the ones where we thought those are most likely to be of interest from a T cell priming perspective. So this is kind of where we are on this trial. It's not a lot of scientific conclusions yet because we actually, for the tumor, only have N of 2 per group. And so we should be getting data back in the next month to expand that greatly. but we do get really good PCR clonotype recovery. I think collaboration with the surgical team and with the pathology is critical, making sure I get a photograph every time and making sure that they actually marked where those samples are coming from. I think no one ever asked them to do a trial like this before, so it did take a lot of explaining of like, what do I mean by these different regions? But they've been great about that. So that's it's been highly informative. And I think we'll get at this question of, can we show that immunogenic cell death actually happened in humans? I think that's my last slide. I could talk more about this. I think when you have all these 4 regions, it's actually pretty easy to figure out sort of among the sharing criteria, sort of who are going to be tumor specific. If you're looking at, either a single biopsy or something before treatment, it gets a little trickier. So I don't have slides, but I can speak to that in a moment. Maybe I'll come back to here and ask if there are questions on this part, or if I should elaborate further on this, there's third point. All right, so we've done a lot of sort of profiling of T cells in the context of clinical trials, and this question of, how do you know that a particular T cell is reacting to a tumor antigen? I mean, we have all sorts of T cells. Most of them are not cancer specific. Many of them are memories, so against our viral infections. Those can even be reactivated. So if you're giving somebody checkpoint blockade, you can get reactivation of antiviral T cells too. It doesn't necessarily mean that they're tumor specific, even when they start to proliferate. And so how you match a particular T cell to a person's tumor is very complicated. So we've been working on solutions to this. We have a collaborator at MIT, Michael Birnbaum, and a shared graduate student who's actually graduating next Wednesday. So I'm very, very proud of him. But to answer this question, and I apologize for not having slides for this, but I'm going to see if I can describe this better. So if you say, okay, if we have a person's tumor and you do still need the tumor for this, so presuming that you can get a biopsy, and it can even be a baseline biopsy, it doesn't have to be after treatment. So, if whatever you used to diagnose the person in the first place, and then you say, okay, if I have peripheral blood, there are tumor reactive cells in peripheral blood. Now you get way more of them if you actually look in the tumor, but even if you look in peripheral blood, they're like 0.1%, right? And so how can you lower the limit of detection to the point where you can actually measure the tumor-specific cells in peripheral blood? We can get peripheral blood very easily. And so if we had an ultra-sensitive method to do this, then it starts to become feasible in the context of a clinical trial. And so what we found is that if you can take that peripheral blood and expose it to the tumor and just say, who's going to react? Now, there are enough dendritic cells in the peripheral blood that they can pick up the tumor, they can activate the T cells. If it's truly an antigen-specific T cell, it will activate. Now, if you're looking at 1 in a million, it's very hard to find 1 in a million activated cells. So what we do at that moment is we say we have a virus that comes in, it uses 4-1BB, which is an activation marker, and it both co-stimulates and infects the cells, right? And so the cells are rapidly expanding and they're green. So it's actually very easy to then say, okay, I'm going to grow these out for a week. I'm going to get them to a large enough group that I can actually find all the green cells, and I can sort those and I can sequence their T cell receptors. And so we've shown that we can take that limit of detection, which, we were by tetramer staining or by other means, you could find them if they were, 0.1%, but if they're 0.01%, no one ever found them. And so we've been able to lower that by a couple orders of magnitude and actually sequence T cells out. So what we've been able to do at the moment is in pancreatic cancer, we actually had matched patient organoids from pancreatic tumor and matched peripheral blood. and just said, okay, random peripheral blood of a patient who wasn't even getting immunotherapy, they just had pancreatic cancer, can we mix those together, add this virus, sequence them, and show that in fact, we were able to expand out all of the tumor reactive cells and show that we could recover those at the end of the day. So I think that is possible. It's certainly something that we've thought now about baking into clinical trials. We have peripheral blood collection is already easy. And so just thinking about, is there enough of the fresh tumor material that doesn't have to go to pathology that you can write into the trial and say, like, please, like, save a little bit of this so that we can use these for T-cell expansion assays. So I think that is currently our best in class method for getting at actual tumor-specific T cells and being able to show that those increased or decreased in the context of FUS. The TCR clonotype tracking that I showed you mathematically gets at some of that same issue, but it doesn't tell you for sure that they're tumor reactive. So there's a little bit of a disconnect there, although this is certainly easier at the moment. Anyway, questions on this?
I was wondering if you could talk a little bit about what you think would or would not apply to brain tumors from this approach. How would that be different?
Oh, I think this is tumor agnostic. I mean, all the tumors that you guys are talking about, thinking about, dying tumors, material still draining to a lymph node. I mean, even in the context of a brain tumor, I think some of the special issues with brain tumors are how do you think about T cells getting back in? And so the trafficking aspect is a little different, but the first question is, do you prime these T cells at all, right? Whether or not those T cells can go back into the tumor is a different question. It's not necessarily something that the original FUS that killed the tumor cell is going to address. Now, it may be something that the microbubble expansion in the vasculature would be able to address later, but I think that's a different question than whether you induced immunogenic cell death.
The PCR clonotype studies, it is useful to do it in mice. I mean, so we can do it in mice just as a proof of concept or understanding if indeed the histotripsy or thermal ablation will prime T cell. But the translation to clinic is more complicated because it's not the same MHC, because it will be different clonotypes. So do you think what do you think about?
Yeah, I mean, you can certainly do clonotype tracking in mice. This is somewhere between $5,000 and $10,000 a person. And so, I mean, for mice, it's just so much, you can use antigen-specific T cells, so why do something this complicated and expensive?
So we are funding a project. The project is starting at VTech where they're going to do that in a pancreatic cancer model. But again, the outcome of the project will be that maybe we can show in this mice pancreatic tumor model that indeed we can prime the T cells, okay? And then what? I mean, do you think there is a clear evidence of that or support that indeed that could be happening in a clinic, or it's a totally different? I mean, we will need to do it in patients for sure. But do you think we can conclude from mice data that the same thing could happen in human, or it's way too?
So I think the mouse data you already have is already good enough to say that it is possible this happens in humans. And so the biggest open question to me now seems not like, can we show this sort of repeatedly in mice, but how do we monitor this in the context of a human clinical trial?
Yeah.
I suppose there's still room to say, like, of all the different FUS modalities, is there one that, induces the most immunogenic cell death and can, after we would do this in vitro culture, you would then go to the mouse model and say, okay, if I do that, do I see that I have the biggest upregulation? But I think the number of things you can test in mice is in the, like less than 10, right? Whereas the number of things you could test in vitro is manyfold more. But showing that it translates from mouse to human is a really, I mean, you guys understand this, right? This is a super important point, and it doesn't necessarily always work out as well. I mean, if you look at TVEC as an example, oncolytic viruses are great in mice. You can absolutely cure them. You can make them refractory to rechallenge. You can cure all of the uninjected lesions, no matter where they are. They could be in the brain, whatnot. But if you look in humans, you're pretty good at clearing the lesion that you inject and maybe some of the other ones in the skin, but it has not gotten to the visceral lesions. So whatever we're doing for T cell priming, it does still seem to be locoregional, not like also clearing brain mets and liver mets. So it's I mean, it still works. It still got approved, but it was less good. And so I wonder, if we'd, tested a few more things in people, would we have been able to get something that is a little stronger of a response? So it's dangerous to say, like, just because we see it in mice, it's going to be exactly the same in
of
humans.
course,
Yeah.
Stephanie, can I ask a question?
Yeah.
Yeah, great presentation. I wanted to evoke a thing like that we noticed when we did mechanical focused ultrasound and double checkpoint inhibition in a neuroblastoma mouse model. Every time that we use mechanical focused ultrasound, so histotripsy type, we noted that there is a very fast decrease of Tregs, and probably because of some cytokine release or so. What do you think? My question is, what do you think is the role of the sterile information that you evoke when you do such things? How can that help in the in this immune reaction?
Yeah. I mean, I'm not sure exactly what's causing a rapid decrease in Tregs that you're seeing. So I mean, I'm sure you're looking into this. In more general terms, this question of sort of what is the dendritic cell doing in sterile inflammation? We want it to go be activated and prime T cells in the draining lymph node, but if it doesn't do that, it's not like it does nothing, right? So the alternative is that it goes in a non-activated state to the draining lymph node and it primes Tregs. And so it's not like you just get less of your good T cells. You actually can swing it the other way and get increased tolerance. And so I think if this is done poorly, right, or and also what's been happening in the patient before they got treatment is that you're building upon a long history of having already gotten a lot of regulatory T cells. And so some of the main problems with cancer immunology is you're not just starting from the beginning, right? You're starting of like trying to induce an immune response where tolerance already existed. And so that's, we can break tolerance, but you're right that there is an existing pool of Tregs that we have to consider.
Thanks.
I was curious, earlier you mentioned inducing that y'all induced autoimmunity in a mouse, resulting in vitiligo. I was wondering if that's like a potential downside of this approach, if it can sort of go too far in having the body react to itself by like sensitizing this process.
Yeah, so that is the flip side of anti-tumor immunity, is that you get induction of autoimmunity. And we certainly see that in the context of checkpoint blockade. I mean, people who are responding really well and they get adverse events, you can get, like vitiligo is not that bad, but you can get life-threatening colitis or myocarditis requiring, steroid cessation of therapy, et cetera. So this is certainly a clinically appreciated phenomenon. It's one that we've gotten much better at managing. That's actually what my husband does as a gastroenterologist. But I think that I would so much rather be in that situation. Fixing autoimmunity versus the person is definitely dying of their metastatic cancer you'd rather be on that end and then working on like how to mitigate adverse events rather than still what are we able to do to this tumor? So I'm not worried about side effects. I would rather see side effects than not. And I don't think there's really a world where you get highly effective anti-tumor immunity that has no side effects, because anything you do to activate T cells is going to have this off target, like sometimes you will go too far.